Good afternoon, everybody. I'm Claire Carvell, and it gives me great pleasure to join Martin Harvey today to talk about the Pollinator Monitoring Scheme. I hope you can all hear me okay. And I also want to extend our thanks to the FSC BioLinks project for the opportunity to tell you more about ponds today. I'm going to start with a quick introduction. I'm going to hand over to Martin for the sort of main part of the talk covering the taking part bit. We'll then have some questions. And then we'll return to me to talk about some of the results and the wider collaborations that we're doing under the whole sort of pollinator monitoring partnership. Next slide, thanks, Martin. So in terms of the context of why we're all here, what we're interested in today, of course, everyone will be well aware that wild pollinators have declined in both their occurrence, so where they're found, and in the diversity of species that are found, and in many cases in their abundance. And this has been happening at both local and regional scales. And for a number of years now, there's been a recognition that long-term international and national level monitoring of both pollinators and the pollination services they provide are really urgently needed to provide the information that we need on the status of those pollinators and on their trends, how they're changing over time. And we need this information from most species in most parts of the world, where simply up until now, we just haven't had that information. The report you see on the right is from the Intergovernmental Panel, the Assessment on Pollinators and Food Production, which actually was published back in 2016. And even just this week, we've had another really nice paper that's come out from some colleagues of ours looking at pollinator declines being an economic threat. And we now have some really clear data on that, how they are starting to affect or could affect global food systems if some of these declines continue. So next slide, thanks, Martin. So this is where POMS comes in. So the UK Pollinator Monitoring Scheme is the world's first monitoring scheme that is generating what we call systematic, so repeated and structured data on the abundance or the numbers of bees and hoverflies and other flower visiting insects. And it's doing this at a national scale. You'll all be aware, of course, that we have some fantastic long-term monitoring schemes for butterflies, for example, and they've generated huge amounts of data and are really telling us a lot about the trends in those insects. But until recently, there simply hasn't been a national level scheme that has looked at bees and hoverflies and all the other pollinating insects in this way. So POMS is combining long-term records of species of bees and hoverflies that are collected by the recording schemes and societies, with these two new large-scale surveys, which we refer to really as the core part of the Pollinator Monitoring Scheme. And those are the flower insect timed counts or FIT counts, and what we call the systematic 1 kilometre square survey. And you're going to find out lots more about the detail on these in a moment. But just to say that POMS really actually, as well as providing wonderful opportunities for citizen science and volunteer involvement and engagement with pollinators, one of our main kind of aims is to underpin the evidence base for the status of pollinators across the countries of England, Wales, Scotland, and now Ireland as well. So we're really underpinning these national pollinator strategies, which now exist in all the countries. And if anyone's interested, I think the links to some of those will be appearing in the chat during the next few minutes. So I think this is where I'm going to pass on to Martin for the fun bit, and he's going to talk to you about how to take part in the two main POMS surveys.
Yes, thanks, Claire. Thanks for introducing the context for the Pollinator Monitoring Scheme. So I'm going to say a little bit more about the how the surveys actually run and some of the sort of statistics about how many people have taken part so far, and to give you a bit of an update on where we've got to with that. And then later on, Claire will report on some of the more detailed analysis that's being carried out as we speak. So I'm sure that some of the people, hopefully quite a lot of the people on the call, will have already had some interaction with the Pollinator Monitoring Scheme, and perhaps you have in fact carried out some of these FIT counts yourselves, but we'll quickly go through how to take part, and anybody who hasn't given it a try yet, we would love you to join in and have a go this year. So as Claire said, we tend to refer to this particular survey within POMS as the FIT counts, which stands for flower insect timed counts. And the idea of this is to gather information on the abundance of insects that visit flowers. And it's a reasonably straightforward way of doing this. We ask people to pick a particular patch of a target flower. So we'll show you the list in a moment. You choose the flower you want to focus on. You define a 50 centimetre patch within which you're going to look at just that one flower type. And during 10 minutes, you count all the insects that actually land on the flowers. And we don't ask you to identify them all the way down to species. That would be quite a challenging thing to do across all the insect groups. So we are asking for the information to be grouped into the main insect groupings. So for instance, we split out honeybees, bumblebees and solitary bees. Then we split out hoverflies as opposed to all the other flies. And then there's a few other groups, including butterflies and moths as one group, and beetles, and some general groups for other insects that don't fit into any of the main groups, and another general group for very small insects. You quite often see sometimes really large numbers of very tiny insects sitting in flowers, and we just lump those into another category for small insects. So it's not providing information on species distribution in the way that the fantastic recording schemes that focus on the species do, but it is providing information on the number of visitors to the flowers within that 10 minutes. And that gives us data on abundance, which can be often a really hard thing to gather for insect populations. And this is one of the sort of new bits of information that POMS is helping to gather. Unfortunately, though, although today is the start of the POMS season, it has been rather cold and unsettled for a lot of us this week. And today the sun is out where I am, but it's still pretty chilly out there with a cold wind. And there are some minimum weather conditions that we ask you to wait for before carrying out one of the FIT counts. So the temperature, if it's a sunny day and the temperature is 13 degrees centigrade or higher, then that's fine for doing a count. But if it's below 13, then really we can't take in counts in those conditions. And if it's a cloudy day, you need the temperature to be at least 15 degrees. So I think we're going to have to wait for a few days longer yet before it's actually possible to start doing FIT counts for this year. But it won't be all that long to wait, I'm sure. And as soon as the weather does pick up a little bit, we would be delighted if you can start adding some counts. And there's no requirement for how many you do or when you do them. As long as the weather conditions are fine, then you can do a count. And we are grateful for every single count, but some people take it a bit further and try and do one a month or even one a week or even one a day in a few cases. And if you can do those more regular counts, that's brilliant, and it all adds value to our data. But there is no set rule to it. It's not like the butterfly transect where you do have a defined day each month that you go out and do it. This is a bit more open-ended. As long as you've got flowers and as long as you've got decent weather, you can do a FIT count between the 1st of April and the end of September. While you're waiting, there's some preparation that you can do, and all the information is up on the UK POMS website. One thing you will need to do is to find a way of defining your 50 centimetre patch. And it's possible to go out and buy a quadrat that defines a 50 centimetre patch, but it's also quite easy to make one yourself. The example at the bottom left here is just using bits of duct tape, and if you fold it over and measure it out, you can construct yourself a tape quadrat. The advantage of that is that you can roll it up and put it in your pocket and carry it around with you. And quite a lot of people use that approach. But people sometimes use 50 centimetre lengths of string and make those into a square, or bits of cane from the garden. There's lots of different ways you can define a 50 centimetre square. But we do need that 50 centimetre area to focus the counts on to give them some standardisation. You can download a recording form from the website and print it out, and lots of people find that a good way to do it. But we do now have an app as well, which I'll come back to in a moment, which allows you to do everything on your phone if you're happy doing things that way. And on the top right there is our list of target flowers, and these are sort of arranged in the order that you're likely to come across them during the year, more or less. And we do ask you to use one of these target flowers if you possibly can, but although they're all quite common flowers, they don't all grow everywhere. And if you haven't got one of the target flowers within reach, then it's absolutely fine to use a different flower. And we do ask you to take a photo of the flower and tell us what it is, so that we can check that we've got the right flower type associated with the count. But again, the more counts that we can get on the target from the target list, the more data we get on those particular ones, and that all helps make the data a bit more systematic. Last year, we launched a new website, and all the information relating to POMS is available from the website now, including the links to download the app if you would like to make use of that. And that did prove to be very popular in 2021. And the app takes you all the way through the process. It asks you sort of step by step to fill in the bits of information that we need. It's got that insect count page where you can just tap on the icon for the different insect groups and add up the counts as you go through the 10 minutes. There's a timer built into the app to measure your 10-minute count. And there's also some supplementary information within the app, including an insect identification guide. A similar guide can be downloaded from the website, but it's also there built into the app. And as I say, we've been very pleased with the feedback we've had from people using this. And in fact, if you look at the number of counts coming in, we did have a substantial increase in the counts last year, which is very, very welcome indeed. So thanks to everybody who took part. But about 2/3 of those counts did come via the app last year. So I think that demonstrates how popular it's been and how it's allowed more people to join in more easily with them submitting the counts. Another thing on the website is we have some information about the numbers that we're getting of different insect types with the different target flowers. And you can go to the website and look at this data and see how it changes as the new counts come in. So the example we're looking at here is for one of the earlier flowering target flowers, buttercup. And this is one of the flowers that you can do a count on. We don't ask you to go out and distinguish exactly which buttercup you're counting on, but if you do know, you can tell us, but otherwise we're happy just to call it a buttercup and add your photo with it. And of all the counts that have been done on buttercup, the average number of insects that people see per 10 minutes is 6. So not too many to cope with. And if you look at the different insect groups, the ones that you most frequently see on buttercups fall into the other flies category in green there, or there's also that large dark grey strip which applies to the small insects, sorry. So in buttercups you will see sometimes some of the tiny pollen beetles and other very small flies and wasps that go into that small insects category, and those are the two groups that are most frequently seen. But you can also see that there is some representation for just about all the other insect groups as well that do at least occasionally visit buttercups. And the chart at the bottom there shows how many counts end up with different totals of insects. So although the average for buttercup is 6 insects per count, in fact the majority of counts in terms of numbers of counts are actually lower than 6. And then we have a few outliers with quite high values where there's been lots of insects. Often this applies to the small insects where just occasionally they turn up in large numbers if you happen to have hit a hotspot or hit a point where they're all emerging at the same time. So the largest number of counts actually have less than 6 insects. So as I say, you're not going to be overwhelmed by insects with buttercups, particularly early in the year. But it's always interesting to see the variety and see how your count compares to what everybody else is seeing. So we'll look at a couple more examples of the different flower types. White dead nettle is another one that is out in flower already, and as soon as the weather warms up, this will be available for doing fit counts. Again, it averages 6 insects per count, but again, it's quite often a bit lower than that in practice. And this is a much more tubular, deeper flower that flies find harder to get into, but bumblebees really like it. And you can see from the large light blue patch there that bumblebees are the main visitor to white dead nettle that we see on the fit counts. Another early example, dandelion is a really important nectar and pollen source for many insects early in the year. And this time our average has gone up to 7 insects, but there's quite a wide range of different counts that people find. This is another one that's very popular. It's another one of these broad open flowers that the flies and small insects find it easy to get into, but you will see bees and other types of insect on there as well. One thing I meant to point out, if you look at the chart at the bottom here with the orange bars, we do get quite a lot of 0 counts on dandelion, and people often get in touch with us and say that they've done a fit count and not seen any insects, and they're a bit disappointed, and they also want to know whether we're still interested in that data. And the answer to the data question is yes, we are very much interested in that. The 0 counts are part of the information that all helps us monitor how many insects are visiting the flowers. So if you do end up with a 0 count, I can understand that it might be disappointing in terms of you've not seen any insects, but in terms of the data, it is still important to us, and we would very much request that you still do send your count in or use the app to upload the records so that we do get those 0 counts as part of the total data set. One more example from the early part of the year with a slightly different arrangement. Again, this is hawthorn, which in my area isn't quite out yet, but it will be fairly soon. And this is a fantastic plant for attracting all sorts of different insects. Once again, we get lots of flies visiting the open flowers, but you will see solitary bees and other things as well. And this one, because it's obviously more of a shrub, you have to sometimes be a bit more imaginative about how you get your quadrat set up to focus on a 50-centimetre area within what is sometimes a huge mass of flowers that you see on hawthorn. But if you can sort of hang your quadrat up, and as long as you can define that 50-centimetre area, you can still do a count on the higher flowers. And the final example I want to show you, because it's a different one, is from the later in the year, towards the end of summer, of course, ivy starts to flower, and we get a very different pattern on ivy. The average number of insects is much higher at 25, and if you look at the pattern of how many counts we get for different totals of insects, it's much more evenly spread across the range. And in fact, we've so far not had a 0 count for ivy, but we do get some very high numbers sometimes. And the orange colour in the sort of donut chart there shows you that there are lots of wasps visiting ivy, which will probably come as no surprise to anybody who's watched a patch of ivy flowers. Wasps do really love them. But we also get a lot of honeybees, a lot of flies, and the rest of the other groups visit as well. So those are just some examples of the different target flowers. If you go to the website and have a look under the view results bit of the menu, there's one that says insects and target flowers, and that takes you to this page where you can then choose the flower you want to look at, and the charts reconfigure themselves to show you the latest figures for those particular flowers. So that's quite a nice little feature for checking up how your counts compare to others. And this is a similar chart to the one we saw before about the number of counts per year, but this time the different colours show you the different countries that are represented. And perhaps unsurprisingly, we get more counts from England than from any of the other UK countries, but we are very keen to get coverage from as wide an area as possible. And particularly for any of you who are based in Scotland, Wales or Northern Ireland, or have contacts in those areas, we can really be very grateful if you can. It's always harder in parts of the country where fewer numbers of people live, of course, but we are trying to get as best coverage as we can right across the UK, and Northern Ireland has more recently joined in with the Pollinator Monitoring Scheme, and we're looking to build up our work there in conjunction with partners in Northern Ireland. But in 2021, we had just over 3,000 counts. If you add all those up in blocks of 10 minutes, it equates to 24-hour-a-day counting for 3 weeks, which is pretty impressive, really, and 39,000 individual insects counted by around 800 participants, all of which is a fantastic contribution, and we are very grateful to see that coming in. But we'd like it to go even higher next year if we possibly can, or this coming year. This is one of the types of monitoring survey that really benefits from having as wide a participation as possible and building up the total numbers that we can use for analysis. So as I say, anything you can do to help spread the word would be very welcome to us. And we hope that people do enjoy the 10 minutes that they spend watching the insects. We do get quite a lot of feedback that it is an enjoyable thing to do and quite a mindful thing to do, to just go out somewhere with some flowers and just stop and forget about all the other things that are happening in the world and focus in on that flower patch for 10 minutes and just enjoy watching the insects arriving and going about their business. And it can be a really relaxing thing to do. And you have to do it in good weather, so you're not going to be out there in the rain and the cold. And you can focus in on your 50 centimetre patch. It doesn't have to be full of the target flower. It's fine just to pick a patch with a few flowers in it. We do ask you to count the number of flowers in your patch so that we can use that to interpret the data. But that's fine to use a patch with just a few flowers in. Okay, so that's a quick overview of how you get involved with the FIT counts, the 10-minute flower insect time counts, and the other major pollinator monitoring survey within POMS we just refer to as the 1 kilometre square surveys. And this is a bit more complex and a bit more involved, and it does involve putting out traps and actually catching and killing insects in those traps. So it is obviously something that is done under sort of quite strictly controlled conditions, and we'll talk a little bit more about that in this next section. So there's a predefined set of 1 kilometre squares that this particular survey happens in, and there are 75 of them. Well, in fact, that number doesn't include the Northern Ireland squares, which were trialled last year and will be running again this year. So the number has gone up, I think, by another 10 to include the Northern Ireland squares. But these have been in place since the beginning of POMS. So we trialled in 2017, and then they've been running every year subsequently. And they are designed to be the minimum number of squares that we can use to detect national changes in pollinator groups and some species. They are also designed to be a mix of agricultural habitats and semi-natural habitats. And you can see from the photos here, some of them are in very agricultural situations with perhaps rather few wildflowers, but potentially lots of crop flowers. Some of them are in wonderful upland locations, some of them are in woodland, some of them are in grassland, so a real mix of different squares. We do sometimes get asked if it's possible to set up a new square because there are only 75 squares, and we'll see a map of them shortly. Some people live a long way away from a square, and to be honest, it's quite difficult to, we do get a lot of requests to join in from people who just do not live close enough to a square to be able to get involved. And it's always a real shame to have to turn people away, but the way the survey works is designed around this set of squares. And because there's quite a lot of resources associated with setting up a square, it's not something that we're currently able to add to. So we are using this predefined set of squares. And what actually happens with the squares is that they are visited 4 times a year. It usually works out once in May, once in June, once in July, and once in August. And on each of those occasions, the pan traps, which are the coloured bowls that you can just see in the bottom photo there, a blue, a yellow, and a white, a set of those 3 coloured bowls is positioned at each of 5 places in a more or less straight line across a diagonal for the 1 kilometre square. These bowls are filled with water and set out between 9 and 11 in the morning, left in position for 6 hours, and at the end of the day, any insects that have landed in the bowls and got trapped in the water are collected into sample tubes, into alcohol, and posted back to the laboratories at UKCH, where POMS is run from. And the volunteers who help us with this go out and set the traps out in the morning. There is a middle part of the day where our volunteers carry out some additional FIT counts within the square. There's usually some spare time as well to go for a walk or go for a cup of tea or whatever else you want to do. And then at the end of the afternoon, the traps are collected back in. But it is a fantastic commitment that the volunteers make to helping us with this. There is quite a lot of walking around and carrying bits of kit involved. And again, it's something that is done in good weather, which we hope helps. And I think people do seem to enjoy it, and we've had some fantastic responses from our volunteers who've been doing this for a number of years now. One thing we wanted to pick up on was part of the 1 kilometre square survey asks you to look at the number of flowers within 2 metres of each set of traps, pan traps that you put out. And the flowers have to be counted. We only ask for things that are actually in flower to be counted, but they have to be linked to their particular flower type. And this also applies to the fit counts. Actually, when you count your target flower, you have to say whether you've counted an individual flower or a flower head. And this is something that people do sometimes get a bit confused about. There is some guidance on the website as to which type of flower applies to the different flower species that we ask you to look for. Do have a check of the guidance or get in touch with us if there is anything that you're unclear about here. But it is quite important that we know, for instance, looking at, I think those are some heather flowers in the third picture along with the flower spike. We're not asking you to count every individual flower, we're asking you to count the entire spike of flowers. And obviously that makes a big difference to how we interpret the numbers, knowing what it is that you've actually counted. So that is also why we ask for photos of the flowers so that we can double check that the numbers do make sense. So once the survey's been done, as I say, the samples are sent back to the labs, and there's a whole other sort of area of work that happens towards the end of the year, whereby the photo here shows Nadine Mishunas, who some of you will know if you're involved with this 1 kilometre square surveys. Nadine does a fantastic job of organising a lot of the sampling kit and dealing with the specimens as they come back in, and there's a big chunk of time that it takes to sort through all the samples. The bee and hoverfly specimens from these samples are separated out into individual tubes, and they're sent off for formal identification to provide the detailed species data that POMS makes use of. And those are sent off to professional taxonomists for that part of the identification. And the other insects in the samples are all counted into their main groups again, but the whole set of sample insects are retained under archive conditions and have started to be used for DNA approaches to analysing these samples, and are being stored in such a way that they will be able to use used for further DNA analysis in the future, which opens up the possibility for identifying a much wider range of species, and also looking at some of the interactions between the species and the pollen that they might be carrying. So there's all sorts of fascinating possibilities for research that can be done on these specimens, as long as they are sort of maintained and stored in the right way. So we're very, very careful to make sure we follow that process through. So if this does sound like something that you'd be interested in getting involved with, we are looking for volunteers to help us with this and to adopt the squares that do not yet have volunteers. And I know you won't be able to see an awful lot of detail on the map on the screen here, but the red squares on here are the ones where we are really seeking additional volunteer help if there's anybody who would be interested in or lives near enough to get involved with those squares. The blue squares have volunteers assigned already, and the grey squares on this map are ones where we have had volunteers in the past. With some of them, we're not quite sure whether they're still available to take part this year. So there's a little bit of uncertainty over the grey ones at the moment. But basically, if you live anywhere near a red or a grey square on this map and you're interested in taking part in this survey or just want to find out more about it, please do get in touch with us. This revised map isn't quite up on the website yet, but it should be by the end of today. So you'll be able to go to the website and see it in a little bit more detail. And as I said, there are now the squares in Northern Ireland as well. So if we have anybody watching who's in Northern Ireland, the Northern Ireland team would be really pleased to hear from you if you're interested in getting involved there. We do have some additional information we can send you about the squares. We've got some descriptions of some of them. This is just picking out the most northerly POMS square right on the north coast of Scotland there. And this wonderful description that Chris Andrews, who works out of the UKCH Scottish office, it's a square that he has visited, and it does sound absolutely wonderful. I wish I lived a bit nearer to it to be able to go and visit it. But they're a real mixture, the 1 kilometre squares. I can't say they're all as beautiful and scenic as this one appears to be, but they all do have their interest and seeing the different insects and plants that you will encounter during the survey is always good fun. Few examples of the insects that get recorded via the pan traps. These photos are not of trapped insects. They're photos taken from iRecord, from other people who've recorded them, just as general insect records, but they're just nice examples of the different species that get encountered. These are all some of the ones that are not seen so often. The moscarda bee is one of our rarer bumblebees, but it does get picked up in a few of the pond squares in all 3 of the British countries. The hoverfly in the bottom left there is a species that was fairly recently added to the British list. In fact, it was only described as new to science in 2013 from Central Europe. And it appears to be associated with pine woodlands. And most of the records so far are from the Cairngorms, and we do have a ponds square in the Cairngorms, which is where this particular species was picked up in 2021. The other hoverfly top right here is a wonderfully stripy species, Hericomia lapona, particularly associated with damp, heathy and sort of bog and moorland habitats. So it's been found in pond squares in Scotland and Wales. And the solitary bee on the bottom right there, the orange-footed furrow bee, is one of the rarer solitary bees that has been picked up in a couple of the English squares. It's a much more southern, warm climate species. And all of these add to the information that is collected by recording schemes such as Bee Wars, and Claire's going to say something a little bit later on about the links between the ponds data, which is collected in this systematic way, and then the more non-systematic data that Bee Wars and others collect. We did a little bit of work over this winter on some of the ways in which our volunteers have engaged with ponds and some of the feedback that we get from volunteers. And these are some initial figures from a report that will be available fairly shortly that Chris Andrews led on with various of the other ponds team contributing to, and picking up on the information that volunteers have fed back to us in previous years, including through the comments that get added to the records. And looking at how many volunteers have been involved in just 1 year, or whether people have stayed over several years. And for the public fit counts, we get quite a lot of turnover in volunteers. A large percentage of participants have just submitted counts in 1 year. That's partly because we had such an increase in counts last year. So all those people who joined last year have only had an opportunity to take part in 1 year, if you see what I mean. So we're hoping that some of those people at least will continue to add counts in future years. But it's the other thing that this, one of the reasons we wanted to look into this, though, was to find out if there is more we can do to provide feedback to our participants and give you more information about what all this data is adding up to. And I think that's something there's a lot of scope for us to do more around that, and encourage people to maintain their involvement and to see how all the information is adding up and what it actually means. For the 1 kilometre square volunteers, the retention rates are higher. There are obviously a lot fewer people involved with the 1 kilometre square surveys, but those people who have got involved do make quite a big commitment to doing this, and many of them have done this for 2, 3 or even 4 years now, and we are fantastically grateful for your support if you are one of those people who taking on the more systematic surveys in the 1 kilometre squares. But again, we know that there's probably more we can do to provide feedback and give people a bit more information about what this is all adding up to. This is just a word cloud from the comments that people add to the survey results that they send in, and perhaps unsurprisingly, there is a real focus on the insects and the flowers and the enjoyment of seeing them and counting them and getting interested in them. A few other comments about the weather and sometimes the difficulty of carrying out the survey in particular conditions, and we will try and take as many of those on board as we can. Some of you may have also participated in a recent questionnaire survey that we sent out to everybody who'd sort of joined the ponds mailing list over previous years. We don't yet have those results fully available, but the initial reports are now being looked at with the ponds steering group, and we will be able to report back on that later in the year as to the results of that. So again, thank you to everybody who took part in that recent questionnaire. And really, these wonderful photos are from one of our volunteers, Tana Holmes, who's based up in Scotland, and has been helping us carry out the surveys with the help of her family. And Catty Baird, who is part of the POMS team who helped set up the surveys in Scotland, met Tana and her family on site and got them all set up. We provide all the equipment and we make the initial contact with the landowner to arrange the permissions. So you don't need to get involved in that side of things. We will provide all that to you if you get involved in a 1 kilometre square survey, and we will set up this initial site visit and show you where all the locations are and make sure that you're happy with the methodology and how to deal with it and how to send the specimens back. So we do try to give as much support as possible to get things set up and ensure that our volunteers are comfortable with what they're doing, but then it's over to the volunteers to carry on the survey in the subsequent months and to keep the whole thing going. So hopefully that's something that a few of you might be inspired to find out more about. But I think that brings us to the end of my section of the talk, and we're going to stop for a few questions now, and then after that Claire will come back and pick up on some of the analysis and results that we're starting to get from all this wonderful data and how that links into some of the other things that are going on. So I think over to you, Dan, if there are any questions in the chat that we can pick up on this point, that would be great.
Yeah, that's super. Cool. So I've kind of categorized questions so we can sort of do them all in together, which should be great. And we've had some questions about doing the POMS fit count. Questions are: do you record the proportion of the quadrat occupied by flowers in that?
Yes, we do. In a fairly broad way, we do. I think there's 3 categories as to whether there are fewer than half the square covered by flowers, about half, or more than half. So we don't ask for a very precise measurement of that, but yes, we do ask you for a measurement of how much of your 50 centimetre square is covered with the target flower.
Magnificent. Vicky asks, how do you map where you're standing? Do you use what 3 words or something similar?
If you're using the app, it will pick up your location from the phone itself using the phone's GPS. So that all happens automatically if you're using the app. If you're not using the app, what we ask you to do is, for instance, if you've noted it down on the paper form or just into a notebook or something, you then have to add your results on the POMS website, and on the website it's got one of the maps. If you use iRecord, you'll be familiar with this because it's a very similar setup, and you can zoom in on the map and click on where your location is from an aerial photo on the map, and that gets us close enough to exactly where you were taking your count.
Beautiful. And Susan is Susan's asking, are you interested in counts on the Isle of Man?
Yes, yes, we do have counts from the Isle of Man from time to time, and yes, they do. Yes, please.
Magnificent. And Vivian is asking, does the app replace the need for the hard copy forms? Does it have all the ability to save or send through the results all via your phone?
It does, yes. So if you're happy using the app, you don't need any of the paperwork, and as I say, the app does sort of take you step by step through. You fill in all the information as you go, and then it gets uploaded back into the main sort of database and website. You don't have to have a data connection in the field. You can do all your app recording in the field, and then when you get home you can use your Wi-Fi or whatever to upload the results subsequently. But yes, if you're using the app actually at the point of doing the count, it will take in all that information and store it, and then you but do make sure you do the upload sooner or later so that the results do actually come into us.
Beautiful. We've got a few questions from Mike which fits quite nicely into this. He's asking, do you record the type of location, i.e. is it nature reserve, public lands, that kind of thing, and do you record the proximity to arable land at all?
So in the app or on the paper form, we do ask you to give a habitat category, and there's some fairly broad categories around gardens, obviously, or road verges, or woodland, or heathland, all the things you might expect, and agricultural land. We don't ask you to say anything about the wider context, but that is something that can be looked at later on in analysis by using existing data about habitats all around the country, and that is certainly something that is potentially of interest, but we don't need you to be making that assessment as part of the 10-minute count.
We've got quite a few questions about the number of POMS counts you can do. Can you do multiples? Can you do of different flowers? And there was also someone saying, can you not do a couple at the same time?
Okay, so as I say, that the fit counts were deliberately designed to be as accessible to people as possible in as many different places as possible. So there is no upper or lower limit as to how many you can do, and some people do lots and lots of them, and it's absolutely fine to do several close together in space. It's absolutely fine to do several on different target flower each time. It's absolutely fine to do several very closely together in time or more spaced out in time. That's all really up to you. In terms of doing more than one at a time, I'm not quite sure what is meant, but what we would prefer you not to do is to use more than one flower type at a time. So when you're doing your count, you should be focusing just on one flower type, one of the target flowers, and pretty much ignoring any other flower that's within your 50 centimeters. It's only one flower type at a time that we want to count on. So that's a definite part of the procedure, is to choose your flower. But having done one, you could then immediately do another one. You could even if your 50 centimeters does have more than one type of flower in it, you could spend 10 minutes counting on one of the flowers and stop, and then spend another 10 minutes counting on the other flower in the same quadrat, if you see what I mean.
Beautiful. Cool. So that kind of answers those questions. I've got a few questions about the results. Can you look at your results after against your own region or just as the UK as a whole?
Okay, so if you go to the POMS website, this picks up on a good point actually, which is quite important to mention. So if you use the app, you have to register an account, and it's free, of course, but you have to register an account to use the app. And if you use the website, we ask you to register an account on the website. And it's quite important that if you are using the app and the website, that you register with the same email address in both places. And as long as you do that, when you log on to the website, you'll be able to see your own data if it's been added via the app or via the website. As long as you've got the same email address in both places, it will all get linked together. So if you have registered and your email is linked up, you can go to the website and you'll be able to see all your own results and download all your own results. You'll also be able to see a map of everybody's counts across the country, and you'll be able to see those flower and insect charts that we showed you earlier on. What you can't do at the moment is to download your results and other people's in the same area. We only enable you to download your own results at the moment. And that is a question that we get asked quite a lot, is whether it's possible to download for a particular part of the country. And at the moment, we just don't have that ability in place. It also raises some questions about data analysis and sample sizes, and whether it's big enough to tell you anything sort of really meaningful at a smaller scale. But it is something that we are keeping under review, and there are often good reasons why people are interested in a particular part of the country and want to get some feedback. And if we can find a way of making that happen, I think we will. But at the moment, it's really aimed at getting your own results out, but also seeing that national context.
Beautiful. And does the info from the fit counts get fed back into the LERCs, night record, etc.?
From the fit counts, it's not directly, and that's mainly because it's not collecting species data, which tends to be what the LERCs and the recording schemes and so on are more interested in. However, the data is being published openly. It's not quite there yet, but later this year, you will be able to download all the data anonymized, so you won't be able to see who's collected it and where they live and all that kind of stuff. But the anonymized data that actually gives the results of the counts will be available as an open data set that anybody will be able to pick up on and use. So it is being shared, but it doesn't link into the biological recording world in quite the same way, because of the fact that it's species group information rather than actual species identified individual information, if you see what I mean.
Beautiful, thank you, Martin. I've got loads more questions, but I think we'll leave them for now, and if we've got time at the end, we can do them then.
Yep, that's great. Thanks, Dan. So and for the next session, I'm going to hand back to Claire, who's going to take us into the next bit, and then, as Dan says, we can pick up on any final questions after that. Okay, so let's just here we go, back over to you then, Claire.
Thank you very much, Martin. So I'm going to spend the next 10 minutes or so just telling you a little bit about what goes on behind the scenes with all the incredible data that many of you have been helping us to generate. And hopefully some of you today will be able to go out and collect some data to share with us. And I'd also like to share with you some of our wider collaborations and some of the impacts that POMS is already having, both locally and a bit further afield. Oh, sorry, I've just... Martin, next slide, please. I just tried to control the slides myself. Thank you. Okay, so I appreciate many of you may be on your lunch break, or it's Friday afternoon and we don't want to spend too long looking at complex models and graphs. But I think, as you know, as a scientific scheme, and because we're doing something which we really want to create a legacy and a data set that's going to be really valuable beyond just the POMS project, I wanted to say a bit more about how the data are used. And again, I appreciate that some of our most recent POMS reports may not be as prevalent or as visible on the website as a few people would like. So this is a chance for you to see some of those more detailed technical data, just very briefly. And then again, if anyone's got any further questions, please do email if you want to find out more. So as again, hopefully you'll all be aware, monitoring numbers of insects is challenging. And so the data sets that we have on insects are complex. Populations fluctuate not just between one site and the next, but of course they fluctuate during the season and from one year to the next, and can be hugely influenced by the weather in a particular year or a particular month. So our aim here with POMS is to be able to detect changes in abundance and in the species richness, so the kinds of species we're seeing or the numbers of species we're recording, and to detect those changes over time, but also to try and account for some of the factors that might be affecting the insects that we're recording. And actually some of your questions this afternoon have already touched on this question about what other bits of information do I need to record to help you understand what might be affecting the insects in the POMS data sets. And this is where we are now creating what we call mixed models to try and pull the data together and look at some general patterns. So thanks, Martin. So what we do is we start, if you think about it, what we're trying to find out about is what is happening to the counts generated within the fit counts, the 10-minute fit counts, or how can we explain the numbers of insects that we're trapping within the pan traps across all of the different sites. So next one, please, Martin. So there are a number of factors that we're able to put into our models of either fit count numbers or pan trap catches. And these are across the board. So we can account for the year, we have the year, the Julian days, the date of the year on which you performed your count or collected the samples, something about the weather in the local location of the survey, so the wind speed and the percentage sunshine, a categorization of the habitat type that it's conducted in, and then the country in which the survey has been conducted. Next, please. For the fit counts, we also account for the flower type. And in this case, so far, we've just been modelling whether the flowers are generally of an open structure versus of a closed structure. We account for the number of flowers you count as floral units in that fit count quadrat. And we also try and account for the context of the patch in which you are conducting the fit count. So whether it's close to or far away from another patch of flowers. Next one. And then additionally, with the pan traps, we also are able to factor in this count of the number of floral units that Martin was talking about within a 2-meter radius of each pan trap station. So all of these factors, all this data that's been collected, are going now into our models of insect numbers, abundance, or of insect species richness that are recorded on the POMS surveys. Next, please, Martin. And this is just a flavour of what some of these models look like. Again, it's not a pretty trend line. And actually, because we've only been running the survey for 4 years or 5 years in the case of now, what you're seeing here are the data up to 2020. We're not really yet able to draw a single trend line through those data points. But what we can do is start to look at both the changes between years. So here you're seeing the total number of insects on a fit count, so total number of insects modelled per count. And we're able to look at some of the patterns that are emerging. So one of the very clear things we see from the fit counts is that the more floral units you have in your quadrat, the more insects you tend to count. Again, that's something we would expect. The amount of sunshine on the counts clearly influencing the counts, so higher counts in areas of greater sunshine or greater sunshine during that 10-minute period. And some other interesting patterns suggesting, for example, that where you've got, again, apologies for the small nature of this slide, but there's no indication that very isolated flower patches are receiving potentially higher numbers of insect visits because they're acting like a honeypot. It seems like most of the counts that are being conducted in larger patches of flowers are attracting generally higher numbers of insects. And again, we can split at the moment habitat types into some quite general categories. We don't really have enough counts in some of the more detailed habitat categories yet, but actually from the public fit count data set, it appears that some of the counts that people are achieving in agricultural habitats are actually returning quite high numbers of insects. If we look next at how that data compares in the fit counts that are conducted only in that network of 75 1-kilometre squares. And remember, most of these squares are in predominantly agricultural or countryside locations. Again, we find generally this one up here that numbers of insects tends to be higher on open structured flowers, very good relationship with the number of flower units in the quadrat. And again, some other interesting patterns my chat's just covering. But we see things like a suggestion that maybe the numbers might have been lower in 2020 than they were in a couple of the previous years. So we'll come back to that. Next slide, thank you, Martin. If we look at the numbers of bees, so just the bees, this is the bumblebees, solitary bees and honeybees that are recorded on fit counts, we see something a little different, and that's here, for example, that bees are generally more abundant on closed, closed structure flowers, so flowers with longer corolla tubes. But of course, what these bars show you is that there's a lot of variation. So it's not necessarily that clear cut. There are many sites where lots of bees visit open flowers and vice versa. But again, a strong relationship with the number of flowers in the quadrat, generally more bees where there's more sunshine on the count. And some interesting patterns here where we see actually that gardens are supporting really good numbers of bees on fit counts, perhaps when compared with habitats that are categorised as agricultural or urban. And also in this case, we had a significant effect of country in the model. So generally the numbers in the fit counts tend to be higher for bees in England. Next one, thank you, Martin. And again, we're looking at the same equivalent, but fit counts conducted in 1-kilometre squares. And with these data, for example, this year for 2021, we're looking to see if we can combine those fit counts so we've got an even more powerful model with more data from a greater range of habitat types across the UK. Next one, thanks, Martin. These are the equivalent models for the pan-trap data. So these are the numbers of invertebrates, the numbers of insects counted in the 6-hour pan-traps that have been set out across the sites. And actually, I think this is my last photo of Richard. I just want to take a quick moment to thank one of our fabulous team members who actually, Richard Dawson, started off as a volunteer on the pan-trap surveys and then enjoyed it so much that he's now joined the team, so works as a part-time surveyor for us each summer and has been introducing volunteers across North Wales. And again, just to really thank Richard and others who are being fantastic ambassadors for POMS. But what you see here are the pan-trap data for all invertebrates. So again, we're able to see, okay, pan-traps in agricultural squares or squares that are more predominantly agricultural seem to be recording higher numbers of insects than those in what we might call more natural or semi-natural habitats. But again, there's a lot of variation here. So that bar shows you that things are very variable. You don't necessarily always get more insects or fewer insects in some of those habitat types. Some interesting effects of year. And again, when we look at the abundance of bees, it also appears that potentially we've got fewer bees occurring in the pan traps in that last year of this model 2020. And actually that echoes some of the results from the fit counts. Thanks, Martin, just go back. That again, we saw an indication of perhaps a drop off in that year. That's fine, thanks, Martin. And again, we look at the different insect groups within the pan traps. So this is the equivalent plot for hoverfly abundance. Again, quite high numbers in the agricultural areas and potentially quite large numbers of species, but not necessarily sort of a stable result. And of course, we know that some of our more interesting and rarer species are still occurring in the areas where we've got semi-natural habitat. So that's the sort of last main model graph plot, but we really just wanted to share with you an indication of the sorts of analyses that we're able to run. Next one, thank you, Martin. So I'm aware of the time. I just also wanted to say that as well as analysing the POMS data in isolation, if you like, so the fit counts and then the pan-trap data, we're working behind the scenes on what we call these integrated models. So where we have the opportunity not just to take trends from one data set or another. In this case, we're demonstrating where you've got the Bees, Wasps and Ants Recording Society, very unstructured, tens of thousands of records of individual species that of course have a very uneven sampling effort because people record wherever they like at any time they wish. And then we can start to overlay these more structured data sets with that unstructured data. And it's already telling us some of these models where we've tried with bumblebee transit walk data from the Bumblebee Conservation Trust, who are partners on POMS. There are signals that our models can be stronger and certainly more powerful when we do combine these sorts of data sets. And of course, as we now come into year 5 and year 6 of POMS with the pan-trap data, we know that we're putting many, many dots on the map here in places that are not typically surveyed for pollinators at all. So we know that those POMS squares are giving us many new sites for many species across the board. Thanks, Martin. Just a quick focus on some of the other questions we're asking within the POMS data set. This is a collaboration with the University of Reading, who again have been partners in the partnership, in the POMS partnership, ever since that we started out back in 2015. And they have had a look within the POMS data set at what is happening with our key crop pollinating bees. So particularly the Andrena ground nesting bees and the bombus, the bumblebee species. And of course, we have a much better understanding now from some of the research data about which bees are most important for which crops. Thanks, Martin. And here's an example where Mike and the team at Reading have just picked out, in this case, all of the Andrena species that are recorded in the POMS pan-traps. This is just from a single year, 2019, where we had quite good coverage, and picked out the bombus species. And we see some interesting patterns here of change. We see early species like Andrena hemorrhoa being replaced by some of the later species. And it gives us an indication that the pan traps are indeed recording the types of insects that we would expect on many of these sites. And hopefully as the data set builds, we will have enough data on some of these key crop pollinators to really start looking at their trends in more detail. Thanks, Martin. Just to finish off, I wanted to, again, just remind you that we're very keen to make the POMS data publicly available. These are a couple of the papers that have been published from our design and testing phase. And as we speak, Martin's working really hard to complete publication of the first 4 years of our core POMS data set. So anyone that contributed data from 2017 to 2020, that data set will be becoming publicly available very, very soon. And in subsequent years, we'll be adding the rest of the data set. So again, making that available to the research community to answer more questions. Next, one of the other collaborations we've had is with the Natural History Museum, and we're looking really carefully at the costs and the implications of being able to use techniques like DNA barcoding on those samples. So do we need to have taxonomists looking at every single bee or hoverfly, or indeed all the other hundreds of insects that we collect in the traps? And with that in mind, we're very, very conscious that these are valuable samples. We archive them and we keep them frozen for, sort of future use and exploration. So every single POMS sample that's being collected is being fully curated and archived. And we've had some fun with, for example, the fit counts were taken up by the Channel 4 programme Jimmy's Big Bee Rescue, which some of you may have seen. That was at the end of 2020 that it showed, and we had great fun across schools and farms and habitats around Peterborough doing the fit counts with Jimmy and seeing how their habitat creation was working. We've also partnered with the Ex-Pollination Project, Learning Through Landscapes, and are really happy to be talking with organisations like the RHS about how the fit counts could be integrated into some of their gardens, for example. Thanks, Martin. And just finally, to touch on the fact that we have a lot of collaborations now that are a little bit further afield. The fit count is being used across all these other countries. Excitingly, we've had a project in South America, Chile, Brazil and Argentina, where they are about to launch the Fit Count app with specific lists that are particular to those countries, and the same for the EU. So the EU are now funding a large scheme to pilot pollinator monitoring across many European countries that have not been covered in this way before. So we're really, really happy to be working with them, and also to be speaking with other colleagues in the US who are looking at the UK system as a model for monitoring bees in particular in the longer term. Thank you. Okay, so this slide is really just a recap. I don't think we have time to cover all of the points, but please do tell your friends and colleagues about your experience of POMS. Let us know what you think. I hope some of you had a chance to fill out the POMS questionnaire. Please spread the word, and please share your photographs or your discoveries on Twitter. And yeah, wait for the good weather was the first point in that slide. Thank you, Martin. I'm trying to wind up so that we don't overrun. There's one more slide. Is it coming? Brilliant. And just again, to reiterate our thanks, we've really been blown away by the support and the interest and the enthusiasm for POMS. And of course, I have to acknowledge our funders and all the partners that have worked with us. Thank you for listening.
Thank you very much, guys. I think we'll have to knock it on the head there because we're overrunning already. So thank you very much. If you've got any questions, I recommend contacting the POMS monitoring scheme through the website, and there'll be contact details there. There's also, we've got some Twitter handles and stuff that should magically appear in the chat shortly as well. But yeah, we'll leave it there. We will put this up for recording, and we'll let everyone know that link as well. But from there, you're welcome to say goodbye.
Yep, thanks very much to Dan and everyone at FSC for hosting this, and thanks to everybody for listening.